Hello.
I can see your presentation.
Yes, hold on, sir.
So good evening. My name is Sheba and I'm from the microbiology department. Today's journal club will be discussing on the research article that is titled Patient-Derived Exosome as Small Interfering RNA Carriers in Ovarian Cancer Treatment. Just a minute. So the overview of the presentation. First, I'll do an introduction on the ovarian cancer burden, where I'll discuss on the prognosis and incidence rate as it stands at the moment. Then I'll also introduce briefly on the small interfering RNA as a system in targeted therapeutics against cancer. And then also I'll discuss MET, which is in full it's an MNG host transforming gene, which is a proto-oncogene, which is usually upregulated in various cancer, and how its role can play, its upregulation can play a role in cancer progression. Then I'll discuss the results, where I aim to answer the question, can knocking down MET that encodes the c-MET, which is the tyrosine kinase receptor, be targeted towards the treatment of tumors, then give a summary to what the research paper concluded. So introduction. Ovarian cancer is one of the deadliest cancers worldwide that affects women, and usually it has various warning signs. This can include feeling full quickly, bloating of your stomach, frequent urination or pelvic or abdominal pain. And although the cause and the risk factors vary from genetics and lifestyle to age, what remains constant is that usually the prognosis is very dismal, because the diagnosis occurs at very late stages. That is when it's advanced in terms of the peritoneal dissemination and also massive fluid buildup. So any drugs that are given are not very efficient in reversing the condition. So with this in mind, treatment strategies have been carried out, including surgery, chemotherapy, and radiation, but still more needs to be done towards targeted therapy of this cancer. Scientists have used for a long time the siRNA, which is the silencing RNA or small interfering RNA. It can be defined as a double-stranded non-coding RNA, which is usually 21 to 24 nucleotide in length, and it's efficient as it helps in targeting or plays a key role in the RNA interference pathway. So the use of the small interfering RNA is great in terms of therapeutics, as theoretically it can silence disease-related genes in a very sequence-specific manner. But the challenge in relation to the use of small interfering RNA is that it's very unstable in body fluids, and also very toxic to non-tumor cells. So to deal with this challenge, researchers have proposed the use of exosomes, as we all know, which are nano-derived vesicles that are heterogeneous in our bodies and produced by diverse cells. And the fact that they have better compatibility in the system and low immunogenicity, and also they can naturally target particular cells or particular pathways, they offer a promising novel strategy towards the delivery of small interfering RNA. So how does the RNA interference pathway work? As shown in the figure, synthetic small interfering RNA can be introduced to the system, usually as a long double-stranded RNA, as we see here, and it's processed in the nuclei by the drosha, which is an RNase enzyme, and then to form a shorter double-stranded RNA that is then exported into the cytoplasm, that is activated or diced, rather cut by the dicer, which is also a cytoplasmic RNA enzyme, to form shorter fragments of RNA strands that are between 21 to 25 base pairs. These are then recruited into the RNA-inducing silencing complex, as shown here, whereby it unwinds to form a guide and a passenger strand, where the passenger strand will be degraded at this point, but the guide strand will be incorporated into the protein RNA complex for target recognition, here where it binds to a complementary targeted mRNA to ensure that the mRNA is cleaved at specific sites, and hence the regulation of a particular gene and also expression of a particular protein can be silenced at this point. So the researchers in this paper aimed at understanding how silencing MET, which is a class 4 receptor tyrosine kinase expressed in various epithelial cells that have been found to play a role in survival, proliferation, and cell cycle progression. But in the research, or as we all know, the overexpression of MET has been linked to progression of various cancers, in our case, particularly ovarian cancer progression. So to briefly explain the pathway, the signaling pathway is activated by MET. MET binds to the ligand, the hepatocyte growth factor receptor. And by doing this, it activates phosphorylation and dimerizes the MET to activate various signaling pathways like the RAS-AMPK pathway and the P13K-mTOR pathway that plays a role in proliferation, migration invasion, and also production of anti-apoptotic signals. So it's very dangerous when it's upregulated in or amplified in cancer cells, as it can basically proliferate and increase the rate of cell cycle progression in terms of increase cancer cells being produced and metastasized. So in this paper, the researchers wanted to use small interfering RNA that worked against MET and knock it down, and answer the question that whether when this is delivered intraperitoneally into mice, can it enable the treatment of this cancer? So the main question we'll be asking ourselves as we go through the results of this research is whether small interfering RNA replacement therapy can be performed with patient-derived exosomes, and by this develop novel precision therapy towards the treatment of ovarian cancer. So I'll move to the results that were found. So prior to starting the cell culture and other experiments, the researchers wanted to establish the expression of the MET in ovarian cancer patients. So to do this, they accessed a database, the JPIA, which is a gene expression proliferating interactive analysis database that has diverse information on various cancers. And they were able to gather data on ovarian cancer patients and also from patients who did not have the ovarian cancer. And accordingly, they were able to establish that indeed the relative expression in terms of the levels of the MET oncogene was very high, or rather relatively high in ovarian cancer patients as compared to patients without it. They also established that in terms of progressive-free survival, which denotes the amount of time a patient starts treatment to when the cancer becomes worse, patients that had a higher level of MET expression had a lower progressive-free survival, as shown here with the red graph plotted. Contrastingly, patients that had a lower MET expression had a higher chance of surviving longer, as shown here with a black line here, that denotes the progressive-free survival. The next experiment they carried out, they wanted to establish what is the expression level of the c-MET tyrosine kinase, which is a receptor of the MET family in various normal cell lines and also in ovarian cancer cell lines. And to do this, they carried out a Western blot procedure where they used immortalized ovarian surface epithelium and F2 as the control cell lines, as expected, because it's a natural a receptor tyrosine kinase. There was expression, but there was higher expression for ovarian cancer cell lines, as shown here from the SKOV3 to the KA8, which are all ovarian cancer cell lines. The researchers then proceeded to design the particular small interfering RNA that was targeting or acting against the MET. And after they designed it, they named it as small interfering MET 3270 or small interfering MET 44. And they transfected this into, rather they first labeled it with a green fluorescent protein, then transfected it to SKOV3 ovarian cancer cell line. And through fluorescence microscopy, we can see the green dots appeared, confirming its uptake in the cells. The next experiment they carried out was a reverse transcriptase quantitative PCR to determine how the mRNA levels were changed depending on the treatment with the siRNA that they developed. Accordingly, as we see in Figure 1E, compared to control, the treatment with small interfering RNA that was designed in both the two ovarian cancer cell lines, there was a decreased mRNA expression, hence showing that the MET expression could be inhibited at the mRNA level and also the protein level through the Western blot procedure that was done as shown in figure 1F, showing control, no change in the c-MET tyrosine kinase receptor, but a change when the siRNA that was designed was used for treatment. So from the first experiment, the researchers were able to conclude indeed the MET is very much highly expressed in ovarian cancer patients, and as much as it's associated with poor prognosis, the use of the designed small interfering RNA had potential in regulating the mRNA expression and also inhibiting the protein expression through the procedures that were carried out. The next procedure, another experiments, the researchers now wanted to synthesize the MET small interfering exosomes that was to be used for the targeted treatment. And to do this, figure 2A, they were able to isolate fibroblasts from patients with either stage 1 ovarian cancer or stage 1 borderline ovarian cancer, meaning that the cancer had not spread to other parts, rather was confined to either one ovary or hadn't spread to the fallopian tube. And so after this, they needed to process it to ensure that it's only the fibroblasts that they harvested. So they carried out in vitro experiments where they isolated the mesothelial cells and the adipocytes. And then lastly, after they obtained the fibroblast, they carried out differential centrifugation procedure and sequential ultracentrifugation procedure in 3 steps to obtain the fibroblast-derived exosomes, which were then, it was then necessary to understand were they really from the fibroblast. And to do this, they carried out an immunostaining procedure whereby they were able to confirm the expression of prolyl 4-hydroxylase beta and vimentin, which are specific fibroblastic markers, and that showed positive presence and negative presence for the cytokeratin 18, which is an epithelial cell surface marker. So this was able to confirm that indeed the exosomes are from the fibroblast and not from any other cell surface. They also carried out transmission electron microscopy, confirming the presence of exosomes which had a rounded morphology, and to be sure, they used CD63 immunoglobulin, as shown in figure 2B. In figure 2C, they did a nanoparticle tracking analysis to confirm the concentration of the exosomes against the particle diameter, which are found to adhere to the proper measurements, which are around 100 to 150 nanometers. So after this, collection of the fibroblast-derived exosomes from the ovarian cancer patients, the researchers carried out a Western blot procedure using a VJ-cell, which was like the normal, used as a control, and primary fibroblast that was obtained from the patients. And they were able to confirm the expression of CD63, CD81, and CD9, which are all exosomal markers, hence confirming the integrity of fibroblast-derived exosome. So with having now the fibroblast-derived exosome, they wanted to proceed to create now the targeted therapy that incorporated the small interfering RNA that they designed and the exosome. And to do this, they carried out an electroporation procedure whereby around 10 to the power 9 exosomes were mixed with a fluorescently labeled small interfering RNA with an electroporation buffer. And afterwards, they were able to collect the encapsulated exosome that had a small interfering RNA, which they henceforth referred to as MET, small interfering exosomes. So with this, they carried out with other in vivo and ex vivo experiments, with the one on the figure 2F denoting the uptake of the FAM labeled small interfering RNA in both the ovarian cancer cell lines COV3 and COV3, hence showing that it was indeed taken up successfully. So in the next experiment, the researchers carried out diverse in vitro experiments to show what was the efficacy of the designed MET small interfering exosome in terms of treating ovarian cancer cells in vitro. And figure 3A denotes confocal microscopy experiments that were carried out after transfecting fluorescently labeled siRNA, that is the 370 or 440. Sorry. Yes. So two cell lines, the SCOV3 and COV3, were transfected with FM labeled small interfering RNA and the far red labeled exosomes, and they were able to be observed under confocal microscopy, showing the presence of the green fluorescent signal that confirmed the uptake of the small interfering RNA, which colocalized with the far red labeled exosomes, hence showing that it was successfully taken up in the system and could be used for the next procedures. In figure 3B, having developed the MET small interfering exosome system and confirming that they'd been taken up through confocal microscopy, they wanted to understand whether they could inhibit or how they regulated the c-MET expression in the three different ovarian cancer cell lines. Accordingly, compared to control, for the three ovarian cancer cell lines, we showed that they were able to show that there was inhibition in terms of the c-MET expression in these cells, and complete knockdown, especially for the CAOV3 ovarian cancer cell line. So they wanted to be sure that this effect was just not specific to the designed siRNA, and to do this, they carried out other Western blot procedures as shown in figure 3C, and they were able to confirm that though c-MET expression was not affected by treatment of either exosome or small interfering RNAs alone, only through the combination of the siRNA that had been taken up with the exosome was the c-MET able to be inhibited or knocked down, as shown in figure 3C. So through this data, they were able to confirm that patient-derived exosomes can function as small interfering RNA carriers. As I introduced at the beginning, hepatocyte growth factor is an important ligand for the interaction of MET that activates signaling pathways that enable cell proliferation, migration, and invasion, and drive cancer cell cycle progression. So they wanted to understand how the treatment of MET small interfering exosomes can suppress hepatocyte growth factor scatter factor simulation through induced c-MET phosphorylation. And to do this, they did a Western blot procedure, and they were able to analyze various conditions and various regulated pathways as shown in this figure. So in the control small interfering exosomes here, after hepatocyte growth factor stimulation, there was a notable increase in terms of the c-MET expression, showing that the hepatocyte growth factor needed to be induced through phosphorylation for it to be active. Contrastingly, in cells that had been treated with a MET small interfering exosome lens, the total c-MET was shown to reduce, and the p-c-MET, as shown here, was absent, hence indicating that knockdown had been done through the treatment with these exosomes, and there was prevention of receptor activation. For most signaling pathways, you know that it's important that GTPases are activated to phosphorylate the molecules. So as expected for the ERK and the AKT pathways, the expression remained constant, suggesting that the effect was specific to the activation and not expression. But for phosphorylated ERK and PAKT, as shown, they were inhibited, suggesting that the MET-C exosome was capable of knocking down the c-MET tyrosine kinase, hence inhibiting these pathways, and also expression in terms of the protein levels as shown in figure D. Further, the researchers carried out other assays to confirm how the MET-C exosome treatment influenced or changed the expression in terms of proliferation of cells. And they did the MTS assay, which is a cell viability assay that measures the metabolic activity of cells after treatment with a substance. And so, as shown here in figure 3A, Compared to control cell lines, we see that the treatment of the MET small interfering exosome 3 to 70 or 44, there was inhibition in terms of relative viability ratio for both SCOV3 and ES2 cancer cell line, suggesting that there was success in the knockdown as the activity in terms of proliferation decreased greatly in comparison to the control that was used in this experiment. Also, the researchers did an in vitro matrigel invasion assay. As we see, a brief overview of how it was done. They treated ovarian cancer cells with the two types of designed small interfering RNA encapsulated in the exosomes, and then they plated it in Boyden chamber that have different compartments, with the upper chamber having the matrigel, DMM and 0.1 BC, and the lower chamber having the chemoattractant. After 24 hours, the cells were allowed to invade, and then non-invading cells were removed and they were counted. So as expected, the treatment with the cells that were treated with the MET small interfering exosomes, there was inhibition in terms of migration and invasion. As shown here, they were less in comparison to control, suggesting also that indeed this system was able to knock down the c-MET tyrosine kinase that drives various pathways towards the mobility, wound healing, and also proliferation of cells, especially cancer cells. These results were collaborated with the ES2, which is another ovarian cancer cell line that showed significant results in terms of inhibition of proliferation through invasion. For the wound healing assay, the researchers also carried out an experiment where they scratched a surface monolayer of cells, and after 24 hours, they were able to observe that the wound healing only occurred in the control cells, but nothing changed in the ovarian cancer cells that were treated with the MET small interfering exosome, suggesting that the migration capabilities of the cells were inhibited by the MET-C exosome treatment, as shown in figure 3G. Collectively, the experiments in this showed that there was inhibition of proliferation, migration, and invasion of ovarian cancer cells in vitro through the use of the system that they developed. So after establishing the capability of the system, they wanted to see how the biodistribution of the MET small interfering exosome occurred in mice in real time. And to do this, they obtained the exosome that was labeled with the DIR dye and then injected it to mice that had previously been inoculated with ES2 ovarian cancer cells. And then they did a real-time observation in 1, 3, and 6 hours after euthanizing the mice. And they were able to show that the accumulation of the exosome was particular towards the peritoneal sites, the peritoneal dissemination sites where the tumor had been inoculated, hence showing that it had a good targeting delivery in terms of going to the particular sites where the tumor had been inoculated, as shown in figure 4B. To confirm whether it was specific, the researchers were able to do in vitro imaging, where the IVIS in vivo imaging use the in vivo imaging system, where they were able to show the localization of the exosome, DIR labeled exosome in the tumor, and after it was removed post-resection, they couldn't see the signal, hence showing that there was targeted delivery pre and post-resection of the mice that had been infected with the tumor. They additionally carried out one more experiment to show the biodistribution, where mice were infected with the SCOV3-13 inoculated ovarian cancer cells, and then treated intraperitoneally by administration of the DIR labeled exosome. And also in this experiment, the luciferase signal was very specific towards the site where the tumor had been inoculated, hence confirming its targeted delivery and success in terms of targeting the sites where the tumor had been inoculated. This was confirmed through other experiments developed, denoted as MET small interfering exosomes, resulted in predominant accumulation to inoculated tumors with successful delivery of encapsulated small interfering RNAs. So the last experiment the researchers carried out was to establish how the MET-C exosome could inhibit peritoneal dissemination on ovarian cancer, and how the tumor volume or the buildup of the fluid in xenograft mouse model could be treated by this system. And in order to do this, the researchers inoculated mice with SCOV-313 ovarian cancer cell line, then which was transfected with the luciferase signal. After which, after around 2 weeks, they injected the mice intraperitoneally with either control, maybe PBS, and also the MET-C exosome 3-70 or 44. And within 35 days, they were checking every week to see the luminescence signal and how it changed. Accordingly, compared to the control, the treatment of mice with the MET-C exosome showed a reduction in terms of the luminescence signal, as shown here. Control, there was no particular change after the 35 days in terms of the luminescence signal. But for the mice which had been treated with the MET-C exosome 3-70 or 44, we see that the luminescence signal decreased, giving an indication that the tumor size was reducing with this treatment of the MET-C exosome. This was supported by data that aimed to see the probability of survival comparatively in the control and the mice treated with the MET-C exosome. The median range of survival was increased in mice that had been treated with the MET-C exosome 3-70 to around 22 days, and the other one to around 24 days. Comparatively for the control, they only had survival to up to 17 days, which showed a good promising strategy towards treatment. Additionally, they were done with the experiments at this point, they also euthanized the mice and wanted to measure the tumor volume over time and also the fluid buildup in the peritoneal region. And the tumor volume for the control remained high, but for the mice which had been treated with the MET-C exosome 3-70 or 44, the tumor volume was seen to have a great reduction. And also the most significant thing they found is that the ascites volume, in terms of the buildup of the fluid, a hallmark in cancer patients, was shown to decrease greatly after the treatment with the system they developed. So through the 5th experiment procedures they carried out, the researchers were able to confirm that MET small interfering exosomes could inhibit peritoneal dissemination of ovarian cancer in the xenograft mouse model that they used. So from this study, the last experiments they carried out was HND staining, whereby compared to control, we saw that the cells which had been treated with MET-C exosome 3-70 were shown to have inhibited c-MET tyrosine kinase receptor activation, as shown here, and also certain downstream signaling pathways, particularly PRK, as shown in figure 5F. Lastly, they did a cDNA microarray analysis, and they were able to show that cancer growth and human hepatocyte growth factor related pathways were significantly suppressed in mice that have been treated with the MET-C exosomes in comparison to the control group. So in summary, for this experiment, they were able to establish that indeed the intraperitoneal injection of this designed exosomes were able to suppress the c-MET tyrosine kinase receptor expression. It was also able to inhibit tumor burden and ascites formation, hence potentially increase the survival time of the mice. With this, the researchers were able to conclude that by controlling the hepatocyte growth factor MET axis, it offered a promising target towards the treatment of ovarian cancer. And the small interfering RNA replacement therapy using exosomes from patient exhibits promising effects for treating peritoneal dissemination of ovarian cancer. So the fact that they obtained the exosomes from patients who'd undergone an omenectomy shows that the exosome could be extracted without difficulty and also mirror the particular metastatic nature of how the cancer cell cycle progression occurs, as it models also the cancer stroma communication, hence better results in terms of the efficiency of the designed MET, small interfering exosomes. So with this, the researchers were able to conclude that engineered exosomes could be employed as small interfering RNA carriers for future molecular targeted therapies towards the treatment of ovarian cancer in personalized medicine systems. So this marks the end of my presentation. Thank you for listening.
Thank you. And I have a several questions to ask the Sheba.
Yes.
I probably ask a simple question, but the number one question is if there's the hepatocyte growth factor receptor knockout, if there's knockout mice with the ovarian cancer, it should be similar effect or no?
Sorry, you're asking if they did a knockout experiment?
Yeah. If
Uh,
there's a if there's a hepatocyte growth factor receptor knockout mouse, or then you should, and then if you implant the ovarian cancer, you think it's you have similar result as you're doing the the SI met exosome experiment.
mm. So they based their experiment on previous research that just showed that
Mhm. So I'm thinking is if this is met SI RNA exosome inhibit the hepatocyte growth factor, then if you knock out the hepatocyte growth factor receptor in mice or knockout mice, should be the similar result. Is it or no?
I believe so, but they didn't do this experiment in this paper.
No, I'm just asking question.
Uh, I'm not sure about that, but from the theoretical perspective, I think it's possible.
And what's the advantage of the exosome experiment compared with other? Because there's other experiment can do similar that can drive the inhibit the HGF, the pathway for different like experiment, like such as like the if you're using the biovector or using the CRISPR to inhibit similar pathway, but if you're using exosome, what's the advantage?
From my understanding, the advantage with this is that they use exosome which offer better pharmacokinetics in the body in terms of targeted distribution of the treatment. Maybe the use of just the siRNA on its own will not be as efficient in targeted treatment. I think so. I don't know if I
And
got the question.
another question, when you see the figure one, and there is a survival rate with the figure one, maybe no, not that one. There's the no, no.
Oh,
Yeah,
this one?
yeah, this one, figure right. Yeah, there's
Mm.
a survival rate. Is it is it really done from the patient or no?
Uh,
Because
they took data from a database that has information on a lot of patients. So they just took a specific lot that was specifically suffering from ovarian cancer. So they just took a subset of the patients in this
there's
database and
no.
compared. Hmm?
So this one is not categorized like which stage, size, or
Yes,
just
yes,
just pulling out the whole ovarian cancer, right?
yes, that's right.
And another question is so what's the key point for this paper? I I know
Uh,
it's this is subjective, but what's the key point? Another key point.
I believe the key point is that the knockdown of the MET, which is an important protein that interacts with hepatocyte growth factor, that has been shown when upregulated in cancer cells can drive its progression. So by knocking this gene out, using the siRNA system that they develop, they can target cancer treatment, as shown in the various experiments they carried out with a xenograft mice model. So the key point is that encapsulating it with exosome offers a better advantage in terms of targeted delivery and the knockdown of the c-MET in an efficient manner towards the treatment of ovarian cancer, which has shown great incidences in populations in the world.
I have a different aspect from this paper because I was very surprised with the result that can get from the figure 5 actually. So why I was a little surprised about this genograph, and it worked in the both homogeneous model, and it also worked in the genograph mouse model. It means, what does that mean? If there's a genograph mouse, it works in a genograph mouse model.
I'm sorry, I don't understand geograph.
Genograph mouse model. What's
Model?
what's the significance of the genograph mouse model?
Geno means different species, graft from different species. That means they use the human ovarian cancer from human, ovarian cancer cell line from human, right? And then human the cancer cannot induce the tumor in the mouse, so they cannot but use immunodeficiency mice. Usually we can use nude mice or the skid mice. So because their choice of animal model in the mice is only two. One is a xenograft model by using immunodeficiency mice. Second one is to humanize the PDGF 뭐라고? PDGF가 아니고 PD. So humanized model is the immune system similar with the recognizing foreign antigen is similar with the human. So the humanized mice model, the antigen can be recognized as self, so no rejection. But PDF model is very expensive, so they just use the immunodeficiency xenograft model. I guess according to their image, they use a nude mice.
Yes,
nude mice is cheaper than SCID. Nude mice only defective in T cells. NK cell and other cells, especially B cells, is normal. So some kind of the immune response can be occurred by the nude mice model, because only T cell defective in nude mice. But if they use skid mice, for example, skid mice, we usually use REG1, REG2 knockout mice model, which the mice do not express the B cell, NK cell, B cell, as well as T cell. So nearly completely block the specific immune response. So they use a nude mice, right? Nude mice is also expensive, but the cheaper than
Prevent
the skid mice. Rag 1 knockout model, Rag 1 or Rag 2.
Mhm.
So usual, and then they monitor after the transfecting the luciferase gene to luciferase, right? Luciferase
Yes.
gene to the human ovarian cancer cell line and injected into the peritoneal cavity, right?
Yes.
After that, 2 weeks after the enucleation of a tumor, they also inject again the cMET siRNA containing exosome.
Yes.
Actually, the specific finding in this paper is that, in my opinion, is that they use the EV exosome from the fibroblast of the ovarian cancer region.
Yes.
That means the fibroblast, even although it is the culture in vitro, but they have experienced some. They communicate continuously with the ovarian cancer cell, right?
Yes.
So they the fibroblast is purified from the omentum of the peritoneal cavity, because the ovarian cancer, usually because of the original location, easily the metastasize to the peritoneal cavity, because ovary in anatomical location just beneath the peritoneal membrane.
Right.
That's why they use omentum of ovarian cancer patients. So
Right.
actually, ovarian cancer itself is all the ovarian cancer at the initial finding metastasized to the peritoneal cavity. So peritoneal cavity, the metastasis means all the cancer cell spread into the whole body, because peritoneal cavity, especially the peritoneal membrane, rich in the lymphatics, very, very highly enriched in the lymphatic vessels. So in usually in the gastrointestinal tumor, when the surgeon dissect the patient, the abdomen, and then he or she find the peritoneal nodule, that means the peritoneal metastasis. Just he closed the wound, that's all. So peritoneal cavity is very dangerous site for metastasis. Nearly all body sites are metastasized by the because peritoneal omentum, peritoneal lymphatics should have a very high the lymphatic vessels. Do you know why?
No.
Why?
Maybe it carries the lower abdominal side organs. It's like a cover for the.
Cover?
I'm not sure.
Because every day we eat food. You eat food. I'm eating every day. So
Right.
when the digestive organ digest the food, then the digestive nutrition should be absorbed by body. So lymphatic vessel needed. That's why the abdominal, the peritoneal membrane have a lot of the lymphatics.
Right.
Actually, omentum is the folded peritoneum. The folded region, lot of lymphatic vessel. End of omentum, the momentum hang the intestine, the intestine, small intestine, large intestine hang over the folded omentum, folded peritoneal membrane. We call that omentum. So the in figure 2, they show the peritoneal fold of omentum. Right, figure A is actually not the ovarian cancer. This is just the fold of omentum. End of this, the intestinal, the small colon or small intestine hang down, right?
Right.
This is a double fold of peritoneal membrane. So this fold by the peritoneal membrane inside this space, lot of lymphatic. So in my opinion, the specific the feature of this paper is they use the fibroblast. Actually not, maybe it can be the cancer-associated fibroblast from ovarian cancer. So the exosome from the fibroblast from the ovarian cancer can easily target it to the in vitro ovarian cancer. And they show that c-MET oncogene is very famous, so not specific. A lot of paper already proved. And siRNA, so actually now the old-fashioned method, not specific. But the fibroblast. So actually targeting is the very, very tricky, usually very tricky. But
Can I ask an extremely simple question? Do you know the reason why acting used as quantitative control?
I know the reason why.
Because every time you're going to ask the question from the defense, right? That question, why you're using beta acting for the quantitative control? Why?
Maybe I'll need help to answer that. I just need to use a loading control, but I don't know the specific
Because
scientific reason.
That's the question every time you will be asked from the final defense, why using the beta acting for the quantitative control? Because I think I explained before, the beta actin is the constant for the cell cytoskeleton, part of cytoskeleton. So because of that, every cell has to be similar. It's not going to be changed because it's the whole it's a construction of the whole structure. So that's the reason why using the beta actin for the quantitative control. That's the very important thing. Every time people ask that one, every professor will ask this question during the defense. Because also other question. This only works for the ovarian cancer.
Yes.
If ovarian cancer with the people who is before the who stopped the menstruation or after after the menstruation. If the menstruation stop, then it's going to be no effect or has effect, because I think the
in the introduction, you already explained about the epidemiology of the ovarian cancer.
It's commonly in people of
age
menopause, after menopause.
So it means you can expect that HGM might be overexpressed after the methylation, after stop methylation, right? Well,
Possibly.
if you just look for this paragraph, it should be because
Mm.
yeah,
Mm.
so it means HGF HGF overexpressed after stop methylation, and probably the reason that has the ovarian cancer expression is have higher rate in age over 63.
Right.
Do you think so? Or no?
I think you're right, but I'll need to confirm a bit later.
Because that's the thing I was see the first from the introduction, so that's the reason I'm asking the question.
Mm. Thank you.
Actually, I knew that ovarian cancer mostly occur in young women, but today I found, according to your introduction, the middle age of ovarian cancer is 63.
Mm.
Yeah.
Uh,
Mm.
but it can also occur in women under the age of 40. So I think it has a median age range these days with the
Mm.
progression of the lifestyle changes. Mm.
Mm. So the very the
Mm.
malignant tumor among the tumors, ovarian cancer is not good. Definitely not good.
Mm.
Mm. Mm. And then ascites. It makes ascites. The first appearance of ovarian cancer is mainly distant metastasis and accompanying also ascites. So this is not good.
Mm.
Initial finding is also not good.
And,
Mm.
mm.
One of the bad cancer.
Mm.
I guess the worse than the pancreatic cancer.
It
Yeah.
was. I thought
Worse
pancreatic.
than pancreatic cancer. I believe that.
Right.
Actually, median survival, 5 year survival. What is the
Maybe if you're diagnosed in stage 3 or 4, you have less than 5 years rate of maybe surviving. That's if it's not completely advanced or metastasized.
but every cancer like that.
Every, you know.
Every cancer.
Mm.
Okay,
Mm. Thank you.
good.
Mm.
Okay.
우리
Good
학생
work.
중에 질문 하나 해주세요. 우리 질문 하나 해주세요. 고은별 학생, 고은별 학생
저요?
질문 하나 해주세요. 아무거나.
아무거나.
응?
소리가 잘 안 들리네요.
바뀌어가지고.
아,
그러면
질문 한번 해보.
김예은 학생. 김예은 학생. 없나?
예은
그러면
학생. 없을 리가 없을 텐데.
송다정 학생, 김예은 학생 있어요? 있어요?
있는 것 같은데? 네. 아마 잘 안 들릴 수도 있어서.
우리 송다정 학생 있어요? 아, 있네. 오, 있어.
네.
네.
질문 없는.
하나만 우리 셰바한테 그래도 한번.
음, 잘 모르겠어요.
오케이. 우리 이소림 학생. 이소림? 이소림? 뭐 없나 보네. 그럼 마치시죠. 네. Sheba,
Yes, Professor.
good presentation.
Thank you for listening and the questions too.
Better
I
than
was a
previous.
lot. Uh,
Okay, thank you.
thank
네.
you.
Thank you. Thank you, Sheba. I was easily to understand what you're actually doing it for your thesis study. Was good.
Thank you.
음, 음.
다음 주에는 제가 발표할 거고요. 제가 발표하는데 조금 아마 여기 계신 분들 아마 저희 학생도 그렇고 여기 있는 학생들도 그렇고 이해하기 굉장히 쉽지 않은 내용일 수가 있으니까 아마 될 수 있으면 쉽게 할게요. 이거는 사실은 최영규 선생님도 인트로덕션 정도는 보실 수 있을 것 같은데 나머지 내용 굉장히 그 바이오 쪽하고 굉장히 동떨어진 내용이라 아마 좀 어려울 수도 있을 겁니다. 아마 AI 분석 쪽하고 그렇기 때문에 좀 어려울 수가 있어서 그 내용은 제가 나중에 같이 하면서 다 설명을 드릴게요. 그래야지 제가 지금 준비한 논문 중에 하나라서 요거는 아마도 하는데 정확히 얘기하면 당뇨발 관련해서 당뇨발 진단기법에 대한 거라 센서를 이용한 당뇨발 진단기법이라 아마 정확히 얘기하면 이제 EMG 가지고 하는 거거든요. 그러니까 근전도 가지고 하는 거예요. 근전도 가지고 하는 거니까 아마 저기 바이오 쪽 내용은 충분히 이해를 하는데 그것은 나머지 공대 쪽 내용은 좀 어려울 수 있어서 그 부분 같이 설명하면서 하고, 그 다음에 지금 그거 맞춰가지고 이제 제가 홍콩에서 발표했던 내용 훔쳐가지고 이제 요즘 하는 아시아 쪽에 관련해서 연구 병양 이런 거 같이 제가 얘기를 드릴게요. 그러면 다음 주에 보시고, Sheba, would you please share your presentation and your thesis for the share folder at Google Drive?
Yes.
Okay. Okay. Thank you. 네, 수고하셨습니다.